All right. Hello, everyone. I'm sorry that I couldn't be here for the first 2 days. And I'm also sorry in case I say some things that already were mentioned. What I will discuss, my talk will mostly be data-driven. I'll be searching for kind of strong fluctuations in actually COVID data, and then tell you how we can actually exploit these fluctuations in order to learn how cases were imported, mostly in England, because sequencing was done to a crazily high degree in England, which was good for research, I think. So let me start. Like, the motivation is going to be on this slide. So if you think about studying the growth of some entity, that could be bacteria in a test tube or viruses as they are spreading through some country. Often the way we model that is like in a mean field way. We say, okay, I don't care about space. This is in a beaker, it's a uniform environment. And then we could say that the rate of change of this, of the abundance of this entity, has something to do with growth, which might be a deterministic part here, where R of t is a growth rate, which can depend on x and lots of other things, of course. But you also always need some kind of noise, which is often demographic noise, where the variance is proportional to the abundance itself. And what appears here is an effective population size, already mentioned by Kavita earlier today. And with that, maybe you can try to model, or rather fit, I would say, something like the rise of the cases, COVID cases over time. But if you're more interested in also dissolving how a variant is spreading, not only in time, but also in space, you need a model that is structured. A simple way to do that is to say, okay, I can decompose my population into different groups that are tightly connected within, but relatively weakly connected between, like cities. That could be cities, for example. So we label them with an index, and then we need here a matrix that describes somehow the exchange of these entities between these populations. And then this equation becomes higher dimensional. We have kind of a vector of abundances. And what is being added here is a term that describes the exchange between these different places. And maybe with that, then one can describe how COVID spread across the world. Obviously, we were, during the pandemic, obsessed by understanding growth, right? Are not larger or smaller than one was a constant question, and people were obsessed by that, and also is a new variant changing this growth rate, which is, of course, an important question. But if you think about these models, there are other important parameters. One is actually the noise. Do we understand the fluctuations in this data? What is it driven by? This is kind of the standard way this is modeled. And another is dispersal. How can we understand the dispersal across space? And this is really needed for good forecasting, but also backcasting. If you have sequenced all the viruses, then you can try to reconstruct how the viruses have spread across space. But to do that, you need some idea about dispersal. And so in modeling efforts, this was maybe measured most directly, but these two other kind of parameter sets were often, just speculated about. There was hardly any measurement, or based on survey data, you could kind of infer something about migration of people, but to what extent does that imply infections, across regions? That requires additional assumptions. So what's urgently needed is a direct view at these parameters as well, especially if you think about future epidemics. So essentially what we've done so far is we try to get a direct way to measure demographic noise and say something about dispersal using the existing data on sequencing of COVID cases. And this is, of course, retrospective data, but maybe it tells us something also for the future. So the data we first focused on came primarily from England, and also not from any time point, but a particular time point. So here you see a time period, I should say. Here you see the community cases. Here's time. And this was done by random sampling. So this is relatively good data, not just biased by how often were people tested. And down here, we see kind of under the hood, the different variants, how they rose. And the region I'm going to be interested in mostly is the span from roughly July 2021 to the end of the year, where the Delta period was, where the Delta variant rose and caused a really high abundance in England, but almost like a plateau. Nearly, not, of course, there are big fluctuations, but roughly fluctuating around a plateau, which to me is still a puzzle, right? Because if you think about simple models are not as either smaller than 1, then it should decay, or larger than 1, then it should grow exponentially. But what explains, over months and months, a constant abundance? There are hypotheses, but for us, it was a great region to focus our research on first. And so this is the region where this Delta variant was prevalent. And the kind of data we look at is so-called neutral variants. So Kavita already introduced kind of what are neutral mutations. They really just act as more or less labels. And those labels appear within each of the major variants. So you see here, for example, let's focus on Delta right away. Within the variants that are all called Delta, you have lots of subvariants, which when you plot them here in what's called a Muller plot, where at each time point, the different slices add up to 1. So they are just frequencies or fractions. You see them just fluctuating here. And you have the same in the alpha case and the pre-B case, and there are some, some arguments on tests one can do. Is this really neutral? So meaning, of course, no mutation is really neutral, but the question is it, is the drift due to, is the bias due to selection relevant on the time scale of observation compared to the fluctuations? And from this data, from these fluctuations, we want to learn something. Yeah. Well, so in this Delta period, as I said, the abundance was roughly constant. Right? You can think about turnover times. The generation time in COVID was about a week. Right? So there are many generations in this process, but no systematic rise here, except for the beginning. You can already read something up, right? You see a lot of fluctuations initially and then less, which is roughly consistent with just having fewer variants initially, more fluctuations, and then less. Okay, so the first part, which is published, in the first part, we just looked at the question, are these fluctuations consistent with the abundance that we also knew for the same period of time, right? So if you really just go through from generation to generation to generation, like in a Wright-Fisher model, you think there should be a relatively simple correspondence between fluctuations and population size. You have the population size. Now let's look at the fluctuations to see whether they are consistent. And the challenge here was that what we observe, these frequencies up here, they're not the true frequencies in the population, because we ultimately just sample the population effectively, because people go to the doctor or are randomly sampled and then sequenced, and this is the data we have, but these are finite numbers, of course. So part of these fluctuations you see up here are just fake fluctuations due to sampling noise, and not true genetic drift that actually is changing the true frequency over time and leading to essentially diffusion of these allele frequencies. So we needed to distinguish that. I'll get later to into the details how we did this. We built a hidden Markov model for that, but that was a challenge. And the main observation was that the noise was much stronger than expected based on the abundance numbers, and by orders of hundreds, so several hundred times, the noise was stronger than expected just based on these abundance numbers. And this was actually the main point here. Another point in this paper was to make a suggestion, at this point just speculation, of what could give rise to strong fluctuations here, even in a period where the overall abundance is roughly constant. That could be that, well, even though maybe the epidemic has reached some constant abundance numbers, we suggested that under the hood, there could be lots of kind of sub-outbreaks happening. How could that be? Just imagine we can decompose the population of England into a population of cliques, right? You have a set of friends, and if none of these friends has COVID, you tended to behave slightly more risky than if you know, oh, a good fraction of my friends have COVID, now I have to be really careful. That's, for example, is the underlying dynamics. You could imagine that if such a clique of friends is uninfected, and a pathogen jumps in, then initially R naught will be larger than 1. It will grow exponentially through this clique until this feedback sets in and maybe it burns out. And so if this is going on, you have essentially sub-outbreaks within these cliques, and the real population size maybe, we have to, that matters here is not the number of individuals, but the number of those. And what kind of this result would suggest then, if this picture is right, that maybe the clique size is on the order of hundreds or several hundreds. But there's actually, so what I'm going to tell you now is preliminary, but suggests that part of this picture might be indeed going on. And that has to do with the side frequency spectrum. So Kavita also introduced the side frequency spectrum before. We look at the newly appearing mutations in these variants, and those that are not fixed, there you can, for each time point, you can measure its frequency, and then you make a histogram of these frequencies, and you find a very interesting distribution. So the blue line is here the expectation that you have if your model is just a right Fisher model going through generations with more or less binomial sampling. Then the expectation is just a 1 over f decaying straight from 0 to 1. Right? This is here has a bend because I'm plotting this slightly weirdly. The x-axis, the frequency is logit transformed, which makes you see the small frequencies as well as the very high frequencies on a log scale. But the real data actually has features at both ends that are inconsistent with this. So first of all, the decay seems to be closer to 1 over f squared. And then you have also an uptick. So a very, very different feature. No decay towards frequency 1, but an uptick in this data. Both are actually indicative, or both would be expected if under the hood we have frequent kind of these jackpot events. Any questions? Please ask questions. Okay, I'll try to explain actually now. I go a little bit into the theory to explain both parts of the spectrum. Well, the first one is, I would like to call it the Luria-Delbrück effect. If you have, and that must have come up in this conference. At least I think in the description of the conference, Luria and Delbrück were mentioned. So if you have an exponentially growing population, and in the course of this growth, a mutation appears, then the mutation will benefit from this growth, especially if it occurs early. Right? So if it occurs early, it's going to be big. So expansions generate large mutant clones. And so just to illustrate what that means, if the mutation happens in the second cell that's being produced, you expect a fraction of 1/2 of the final population to be mutants. If it occurs in the third cell, you expect 1/3 of the final population to be mutants. And generalized, if it occurs in the nth cell, you expect a fraction of 1/n of the final population to be mutants. So that suggests that the probability that the mutant fraction of one mutation entering this growing population to be larger than little x should go like 1/x, or the probability density to go like 1/x squared. Right? This is a funny marginal distribution where not only the variance diverges, or is dependent on the cutoff, but also the mean is divergent logarithmically. And these jumps or these very large offspring number events, which we were called jackpots, they could plausibly be responsible for this 1/f squared tail for low frequencies, and would be expected, this kind of clique picture also is the case. But then what about this other tail up here? This actually tells you not something about jumps, but something about a systematic drift in your dynamics. And such a systematic drift is expected if selection is active, because then variance would be pushed up or down. In this case, it looks like being pushed up due to selection. But actually, if you have such a broad offspring number distribution, there are sampling biases emerging that look like selection. But it's really just a sampling bias. I try to illustrate that using kind of a generalization of like a Wright-Fisher model, which allows you to study any kind of offspring distribution, and we look at the broad offspring distribution I just mentioned. Imagine you have n individuals and a fraction x at time t is red. And now the next generation is generated as follows. Each individual gets drawn a family distribution, or offspring distribution, from the distribution you're interested in. We are interested in a Pareto distribution, like that. Typically, if you do this for the entire population, your total population will be way too large. So you downsample, you binomially downsample to the old population size. And so now you have a new fraction of mutants. You go from xt to xt plus 1. And so in this way, you can ask, what does such a broad offspring distribution do to the frequency dynamics? And one effect I really want to highlight is there will be an effective sampling bias here. So, and you see this already if you just go from one generation to the next. So let me illustrate this here. This is in this kind of little computer experiment, I started with a frequency of 1% for the mutant, so very, very low. And then drew from an offspring distribution with this parameter alpha equal 1, the 1/x squared distribution. And you see that the new generation is, of course, a random variable, but its histogram looks like this. What you clearly see is that there's a peak, and the peak is displaced from the 1% you start with. It's actually at lower frequencies. So there's a apparent bias, and if you start so here towards lower frequencies, if you start with a very high frequency, above 50%, the bias goes in the other direction. So as if the rich always get richer. How could that be? Because the model that I created was a clearly neutral one. On average, indeed, the average of the next generation's frequency has to be exactly 1%. How can that be true with this kind of skewed histogram? The reason is they are rare compensating jumps. Right? Rarely, from this 1%, you can jump close. You can jump to the other side of 50%, to a very high frequency. Those jumps exist in the plus direction, the up direction, but not in the down direction. right? And those happen extremely rarely, but they rescue kind of the average to be what it was in the original, in the generation T. So rich get richer, but rarely the poor can turn the tide. These days, I think there's a perfect metaphor for the US and how people live there. Jackpot events. Yeah. About the US? The sample size is 800 here. 800. So you do see the poor here that are turning the tide. They do power up. That's what keeps the mean. That keeps the mean indeed at 1%, right? So you have kind of a typical, I would say this, a typical behavior as we understand in physics, but I think you can make it rigorous. You move in this downward direction, so towards, the rich get richer. And that's indeed kind of a bias. It's a drift term. But you also have a jump term, and this jump kernel has a finite mean. And and we can be, how you can estimate this bias for the majority. It's actually not hard. So we draw the offspring numbers from this reverse cumulative distribution. If you have a finite sample, you necessarily have a largest drawn offspring number. So you kind of can think about the histogram for, let's say, the wild types that I draw to kind of separate out this reverse cumulative distribution for low offspring numbers, but then more or less it's cut off at the largest frequency. Now the mutants have a smaller frequency, so they sample fewer offspring numbers. So the cutoff is smaller. And that has a consequence for the kind of typical mean you expect. Yeah. So now if you compute the typical offspring number by integrating this distribution all the way to the cutoff, you pick up a cutoff dependence, which you wouldn't do in a normal distribution with finite mean and variance. Here the mean is logarithmically divergent. That's why you pick up this logarithmic dependence on the frequency, which results in what looks like a fitness effect, but it's a frequency-dependent fitness effect due to the sampling bias. And this can be extended to other alphas. We've done this in a recent paper where this bias actually becomes time-dependent. There's a time-dependent, alpha-dependent power law appearing. You can ask me later if you're interested. But the point is this drift, this rich get richer effect is what we see in this tail towards 1. There's a piling up of variance at high frequency because this drift is slowing down as you go towards higher frequencies. And this generates a tail that has this mathematical form. We actually not only have the asymptotics, we have the exact universal side frequency spectrum for the situation. And it seems to work quite well for SARS-CoV-2. Yeah, I agree. So the actual selection that you would pick up here would be a logarithmic one. So if you can resolve this logarithm, then it looks like all the variance that we are studying here would be under a similar selection. You might say, okay, I can't see the logarithm here. That's fine, but all of them roughly would have to have the same selective advantage to explain that. But I don't have kind of that's all I can say. I can say that a very simple theory fits. I would say that, yes. And also here you see it for two variants. This is for the alpha period, this is for the delta period, England, US, Germany, Denmark, Japan. And you see now the dashed line is actually the true exact distribution. And well, it's always different from Wright-Fisher for sure. And I would say pretty consistent with that. This one? Oh, this one, no, yeah, those details I don't know. Well, there's of course also the situation that for the side frequency to really be observable, you need to have enough time, right? To actually, we're talking about mutations that first appear at low frequency, then they have to reach these high frequencies. And so you have to have enough observation time. So this could be a finite time effect. Another study, another control we've done is we focused on mutations. But part of this could be, there's of course exponential rises in these variants. And maybe some of these jackpot events come just from the overall growth of these variants. So we also looked at variants that just appeared in the plateau period, so definitely did not benefit from the initial rise. And for those, we have just not enough time, but we definitely see the 1 over f squared. There should be 2 peaks, you say? Seem to be 2 peaks. I don't know. I haven't. Okay, we can look at this later. So I don't know. At this level, I can't tell. So this is very preliminary, I should also say. How much time do I have? Okay, let me actually be quick on this one. It's also not so much on rare event statistics, but I think I'm super excited about this. So all these fluctuations, they generate frequencies that are very different in the different regions. If you look really now at space. And so we realize that perhaps we can learn from the relaxation towards a common frequency, something about how variants are being exchanged between regions. So, and the basic idea is illustrated here. So imagine you have 2 regions, A and B, that first are in a lockdown, and at first the frequencies of a variant are very different in these regions. So in A, the variant has a high frequency, in B, the frequency is low. And what do you expect in the presence of a lockdown? Well, these frequencies should just fluctuate around their original value, perhaps due to jackpots, but there's no reason for them to become similar. But once you turn on or turn off the lockdown, they start to exchange variants. Here, assumed that A gets more imported cases from B than the other way around. Then the frequencies should come closer to one another. Still, of course, we have fluctuations, but from the relaxation to a common mean, you can kind of learn how strong these couplings are. It's just like a Brownian particle essentially being trapped in a harmonic trap, and from the fluctuations, you can learn the stiffness. This relaxation process can be seen in real data. I don't have the time to go into the details. Here's kind of the theory for that. The frequency in the next generation in region I of a given variant is expected to be the same frequency as in the previous generation, except for the importation due to noise, right? But noise is on average 0. But what do importations do? Well, each one of these each one of these terms describes the importation from J to I. And first of all, the exchange between J and I will only have an effect on I if the frequencies are actually different. Right? If they are the same, then exchanging them, you don't expect the frequencies to change. So that's why the difference appears here. And how strongly the importation is from J to I is encoded in this matrix element. It's essentially, if you average this, a linear equation. You can fix the diagonal to make this a right stochastic matrix. It has a nice interpretation in terms of importations and also backward in times in terms of a jump kernel between the different regions. What do we find? Okay, this is how we do it. We do a hidden Markov model. But what you find is a kind of a big matrix of interactions between different regions, which here is already ordered in such a way that the rows are as similar as possible, neighboring rows are as similar as possible. And then you see that really regions that you expect to be close together also are close together in this matrix. For example, here you have a big block of London, suggesting that Well, they are strongly coupled. So roughly, these blocks appearing, diagonal blocks are appearing. We also see that the interactions decay systematically with distance. The closer you are in physical space, the more you exchange variance. Apparently, the faster is the relaxation towards a common mean. And I find that really cool because, right, we don't put in any spatial distance. It just comes out from looking at these fluctuations. We didn't have migration data, mobility data in England, but we did have it for the US. And there we see that the decay of our interactions, kind of the epidemiological interactions, is slower. So this is a blue line, than the exchange of people between these places, right? Which suggests to me that indeed there's a systematic difference between just looking at mobility and the true epidemic interactions. And one possible explanation, pure speculation, is that long-distance travelers, they tend to have stuff to do in these places they are visiting. So they behave a bit more risky. They tend to be more infectious. That's why the decay with distance is slower than expected, just based on mobility, how mobility flows are. There's some variation of this matrix over time, but I want to come to an end. Just summarize that, okay, allele frequencies in this epidemic context fluctuate really excessively. It's not well described by Wright-Fisher noise. Seems to be consistent with broadly distributed jackpot events. It's preliminary, possibly due to clique structure. And from relaxation of fluctuations, you can learn importation rates. And we hope that with that, perhaps we can build better models for the future. With that, let me just thank people who did this work, mostly Takashi. He was really instrumental, is now professor in Tokyo. And Shinshin worked on the strengths of noise. She's a postdoc at Harvard right now. All right, thank you.